So, hi, my name is Brandon Shaw, and I'm the Clinical Cytogenetics Director at Henry Ford Health System. And today I will be talking to you about the analysis of optical genome mapping data in VIA. And so the agenda today, we're going to go through some of the features in VIA, so pre-analytical analysis, and then the analysis, and then as well as how to report the variants and things, and how to generate reports straight out of VIA. So this is going to be a live presentation, so I am going to be within our VIA system during this presentation. So some of the features that I would like to point out just, very quickly, as an admin, you do have the ability to go in and kind of structure things so you can add sample attributes that you want to show up in your home screen in this tab. You can do a workflow. So right now it's where our workflow is set up for a Tech 1 and a Tech 2 review and then director review completed. You can make this as complicated or as simple as you'd like, just depending on what you want. The event classification system. So our event classification system looks like this. These are also basically kind of recommended by Bionano during the setup portion where you're working with VIA. You also have decision trees that you can put in and modify. So this is a decision tree that it classifies events based on, just an algorithm basically. And again, Bionano works with you very well to give you these classification trees and then help you edit them to kind of fit your laboratory's needs. Sample review preferences. This is where you can kind of, and you'll see this in a minute, but with your tracks or filters or whatever you want to set up that way. Gene panels, so you can add pretty much any kind of bed files, gene panels that you want into your analysis. We use these as filters to kind of go through some of the samples based on guidelines to find relevant changes. You can also, if you'd like, have guideline-based analysis. So they do provide you with AML guidelines, PanHeme guidelines. We prefer to just have our guidelines set to the PanHeme when we do ours. And then you can add any kind of report templates that you would like to generate out of VIA 2. All right, so now that that's all done, we can go back to the home screen. And here it's got a really nice feature where if you put these stars, you can kind of type in anything, and as long as it's surrounded by stars, it will pull up any samples. So you can see here I typed in OGM, and I've got 3 samples that pulled up with OGM in the name. For the point of this talk, we'll be analyzing OGM1, and then we'll pop into OGM2 just for a brief moment and a little bit later. So, all right, so then you just simply click on that. It'll open up the case here. You can see that I have 2 windows that are split out. This first view is the post-analytical view. So the whole point of this view really is to try to determine if there are any large copy number changes. And the best way to do that is you can see you can go through and look for these red and blue bars. This sample is pretty quiet as far as copy number changes. You can also use your whole genome view so that you can see all the SNP probes down here at the bottom. Again, there's nothing really indicating that there's a large copy number change in this sample. And you can also click on this nice aneuploidy tab, which goes through and lists all the chromosomes and tells you if it's seeing anything associated with a copy number. And again, in the sample, there's not really any of those visible. So then we would next go into view 2. View 2 is a small guidelines view. And so in this view, we're just kind of looking at we want to go in and look at our small guidelines and our gene panels that we have, right? And so to do that, it generates this table, right? This table is going to be really busy for this because there's a lot of changes, but then you can click on panels. I do know that this sample was associated with acute lymphoblastic leukemia. So then we would click the ALL small variants panel, which then pulls up all of our little variants that we know are associated with ALL, and then modifies your table so that you're only seeing the things that are in your sample that are, have those or in those regions. So you can see here that soft CNV mask overlap. So this is actually your decision tree at work. So this is pre-classified that it overlaps with a region that is known to be a problematic area. And you can also see, and you can adjust these windows down, but you can see in your tracks here where I've got all these things separate out, but your soft CNV track is right here. So soft CNV mask, these black bars are regions where we know that they're a little bit noisier than what you would anticipate. So these are kind of filtered out using your decision tree. The things of interest in this panel, so obviously this was a tier 4 finding. but you can clearly go in and check it if you'd like to as a director and kind of go through and look at your structural variants. This is a tandem duplication. Also shows up in OGM2, which is a new feature in the newest version of VIA. And as you scroll down as well, you can see that you have your DGV track, and this little duplication area has been reported in DGV, which again is one of the reasons why you classify this as a tier 4 on top of the fact that kind of a common thing that we see a lot in the lab. This one is classified as a tier 1A review, so you would want to kind of pay a lot of attention to it. And you can see that this is in IKZF1. It is a deletion. And we know that IKZF1 deletions are associated with ALL. And if you wanted to, you could again do your little drop down, and you can see that we've also seen this in our OGM4 sample in a little bit of a larger form. But again, it's something that we've seen before. One of the other things too that are nice about OGM is that you can also have your knowledge base. And so you can see here that in our knowledge base, we have that there is an IKZF1 deletion, including exons 4 through 7. And if you wanted to, you can click on that and open it up and go through and kind of add interpretations and those types of things, which will be important a little bit later in the talk when we get to view 6. So for this one, I would classify this as tier 1A. In order to do that, you would have to start editing the sample and click on the little pencil here, which would then allow you to change things. So now it's a tier 1A finding. And other than that, this is pretty quiet in ALL small variants. The other thing that we would want to go check, though, for our panels is our Bionano cancer gene list. This is a nice feature, this window that pops up. So apparently some of these genes that we have in our panel have been moved. So it's just kind of a warning that you need to go update your gene list. So again, VIA kind of takes care of you from that standpoint. So going that, but other than that, you can see that as far as the cancer genes, there's not a lot going on in the cancer genes, and we've kind of already all classified these things based on those on our classification table. One other thing that I would like to add is that if for my access people, because I am not really one of those, but because I've been doing analysis in VIA for a long time with microarrays and now incorporating OGM, but if you wanted to, you could link out to access and go into browse region and go into access, and it will open up access in a separate window, and it will go in and open up this particular variant and go straight to the region where it's located at. This would be from your sample map, and this would be your reference map, and then you could also right click, open up, show molecules, and just kind of go through and look at those. And then easily pop back in the VIA to complete your analysis. So the next step that you wanted to do is the ONC3 that would be large guidelines. So the purpose of this is to try to go through and look for large copy number changes like whole chromosome changes and those types of things. We do have panels that are associated with this. Again, you have your ALL large variants panel for chromosomes 4, 10, 17, and 18. Obviously, this sample's pretty quiet from that standpoint. So nothing to see really here. Then the next we would go to would be ONC4, which is your 200 kilobase variants. This view allows you to kind of look for things that may not have been in your panels that you may have missed, and you can see that there's definitely a lot more things in this table. Although again, still this is pretty quiet, but you can look and see that most of these have been filtered out by our decision tree as in soft CMV mask overlap regions or things that we had already classified. The next view is a little bit scary just because it is the negative check view. So this is where you want to go through and make sure that you didn't miss anything in the course of your analysis. But the way we use this, because there is always a lot of things right in here, is we have a panel that we use called the manual check panel, which is already applied here. So if you take this off and go to no panels, you can see really quickly that your list of things to review gets really large very quickly. But if you apply just this manual check region, and again, I'd also like to point out that most of these, though, have already been pre-filtered out by our decision tree, right? Where there's a solved CMV mask, or if they're in the control database, it kind of labels those for you so that you don't necessarily have to go through and review those. But what we like to do is have our manual check panel that we have that is available to us. And so this is just kind of to go through and look at regions that we know that are kind of difficult. And so those include IKZF1. And so it's really simple from this standpoint. Once you have your panel open, you can click on IKZF1 and it takes you right to that region. And as you can see, this is the structural variant that we've already classified. But we also know that there's difficulties with CRLF2, and so you can review that region looking for your CRLF2 gene rearrangements that occur. IGH, IG kappa, IG lambda, those regions are historically difficult no matter kind of whether you're looking at microarrays or optical genome mapping data. So we just want to go through in those regions and make sure that there's not really any kind of translocations with chromosome 14 to a different region. And once that is all done, you would go to the director review, which is kind of where you would start off as the director once your techs have finished all the analysis. This table is just kind of everything that they have classified so that you can go through and review it and make sure that you want to include it in a report and add your interpretations and those types of things. So for this one, you would simply check there, which means that I want to include this IKZF1 panel. And when you're looking here, I know that I talked about the knowledge base earlier. So the way we kind of use our knowledge base is if we have this here, we have our interpretation for this particular deletion. You can very quickly copy that and put it into this interpretation window in your table. Once that is done, it will auto import into our report in just a minute. But before we do that, we want to click back out of here, go to our Circos plot, and we want to annotate this, right? So we want to show that IKZF region is here, so IKZF1. Once we do that, we can click this annotate button, and then it'll label where IKZF1 is in the red bar, indicating that deletion. From here too, I would want to click on these just to kind of clear out everything else that I don't want to show up in my report. And from there, everything else is pretty much done. You would go here to your report drop down. We have just a general heme template that we use for most of these samples. So you click that. It'll ask you to open it up where you want to save it. For this, I'll just save it to my desktop. And then, yes. So then it creates the report. The reward report comes out. For our purposes, when you build these, you can have, I forgot what they're called, but basically you can put in windows and stuff into the Word document where you can have it auto-populate from what's in VIA. And so you can see that our interpretation auto dropped in for this IKZF1 deletion. The way we structure our reports, we would then modify this to say it's positive for IKZ loss. And so that is there. There are no section 2 tier 2 findings. And so we also have it set up where our Circos plots auto drop into our reports. You can see that IKZF1 is here. And we also include a whole genome view in the reports, as well as our table that is auto-populated, showing what we found. And then your methodology. So these reports are very nice. And then from there, we would actually save this as a PDF, which we can have auto-dropped into our LIS system and reported in Epic. So VIA makes all that very simple and very easy. So before we get too much farther along at that point in time, you would want to change this to your director review completed, quit editing, go to this sample, which allows you to lock the sample. So that basically locks in all the databases and things that you have utilized in the analysis. And so I can say, just real quick to show you that. Actually, I need to drop it down to here. So when you're looking at your tracks, a lot of these tracks are provided by OGM to you. Other ones, like I said, you can modify and put in bed files. So for our samples, we have a lot of different tracks that we use. So we have our pan-heme track, our NPN guidelines track, your ALL guidelines track. You have your civic variants and civic genes that you have that are visible to you. Any kind of segmental deletions, duplications, those are available. Your knowledge base tracks are also there for you to review those things that are in your knowledge base. You can also, and we have a bed file that's not included in this version of VIA yet because we haven't quite got done validating it, but you can also add cosmic genes if you wanted to from a bed file, or a DGV gold bed file if you wanted to incorporate that. So everything's really easy to do from that perspective. So that's really all I wanted to talk about with this sample. Moving into a second sample. So this is OGM2. So this sample is an APML. And the reason why I wanted to show you this sample is more to kind of go through like what a translocation looks like in VIA and how you kind of look at those. It's a translocation in APL, so it's a 1517. You can go to your table, go to small guidelines, panels, put in your AML small variance panel. You can see here that there's a translocation between 15 and 17. The way this shows up in VIA, and for Access people, I know you can link out to Access from here too, and quickly pop in and look at this translocation data in there. So we can do that very quickly just by zooming in. There we go. Browsing region, Access, and again, Access will open and you'll be able to see your molecule files and everything from Access. Which allowing you to see that, yes, this is indeed a translocation between chromosomes 15 and 17. But coming back into VIA, so how do you know this is a real fusion? So you can see here in the table, you have your fusion junction 1 box, right? And you can see from your gene list here that they've provided that PML goes in that direction. And then when you click over to 17q, it jumps right out to where this other end of this translocation is, and you can see that RAH also goes. So both genes are going to the right. You can see your fusion junction here where it's got the arrows pointing in as well as the arrows pointing out. And so Bionano has given us this lovely little table that allows us to kind of assess these fusions in a manner where if I know that my fusion junction annotation gene types, right? So the arrows are going towards the middle and my arrows are going out. So this is that situation. I know that my genes along the P to Q reference are both going this way. So I would be looking in this top box here and in this one here for the ones where they're going in opposite directions. The way this A to B is set up is the lower chromosome would be A. So chromosome 15 would be A, chromosome 17 would be B. And so based on my fusion annotation junctions as well as my gene directions, I know that I am in a situation where one fusion results in A to B, the other fusion results in B to A, where 15 and 17 are hooked up together, indicating that this is a true fusion. If you had like a junction annotation or your genes were in odd directions that didn't quite map up with your fusion junction annotations, then that would be an indicator that this potentially is not a real fusion. One other thing that I wanted to show you real quick is kind of how to assess copy number variance, because we didn't really get into that very much. So this sample here, when you open it up, you can see that there is obviously a translocation between chromosome 7 and 8 based on the fact there's loss of 7p and a gain of chromosome 8. It would suggest that there's a DIR 7-8 translocation. When you zoom in on the 7 region, you can see that there is the corresponding copy number event that is there. If for some reason, and this does happen rarely, but it does happen on occasion, you were not happy with the size of this translocation, so you thought maybe it was actually, this deletion, you thought it maybe was a little bit larger, you can go into edit mode. You can select this little selection tool button here. You can click on your CNV event and hover right around towards the end of it, which will give you these double arrows so you can kind of adjust the size of that deletion or snap to the probes once you get ready to do that, and it'll kind of, and then if you want to, you can run the auto classification for the purpose of this. I'm just going to say no. Another thing you can do is say if you wanted to draw in a deletion, you can simply put your crosshair arrow here and kind of draw that in there and call it a deletion, a gain, a loss, amplification, whatever you want to. So in this case, we could call it a loss, and you could snap to those probes and it would draw that in for you. And if you made a mistake, you can also go in and delete it. All right. So I think that I've gone through most of the things that I wanted to show you in VIA. I will say that the software is very friendly to use. There are a lot more complex cases that you can have, and I feel like VIA is very good with the decision trees. So this OGM4, so you can go in and have this little information tab. And then based on that, you can enter the name, OGM, you can enter the date you received, which for this we're to say 1/1/2026. If you have the session numbers in your lab, you can include those in here. One feature I do like is that you can separate things based on the WHO cancer types, which is very nice because it allows you to go back in and build almost like knowledge bases. We're going to be switching back over to the live view now. And one of the other features that I wanted to show you in VIA is two things really. This multi-sample view, where you can have all the samples of interest in one area and kind of go and separate these things and look at your SNP probes or your copy number probes and very easily look at this. So that what we're seeing in this window is a group of CNS tumors, and you can see kind of the copy number profile across multiple samples. The other thing that is very useful that you can do in VIA is when you're analyzing a sample, you also have these nice bars up here at the top where you can generate a global similarity plot. And so once you have enough data in these databases, VIA will actually tell you that your tumor copy number profile is more similar to, in this case, a metastatic melanoma compared to a glioblastoma. So I think this global similarity plot is also another very useful tool that you have at your disposal. I'd be remiss if I didn't mention the people in the lab. So Dr. Alice Fang, who did all of the validation studies for OGM analysis, Jessica Sanchez, Marty Harvard, Rondi Dotson, are all techs in the laboratory who are heavily involved in the validation of OGM and getting all these samples to run smoothly. So Jess particularly is instrumental in our ability to get all these things done. Nancy LaFlave is our manager in the lab. And then Natasha Diaz-Meyer is from Bionano, but I will say that she takes time out of her week every week to meet with us and just to kind of go through things and make sure that we're not having any questions or issues, and has been very, very helpful with making sure that we stay on target and any kind of problems that arise. She's been invaluable in getting those done and taken care of, and helping to explain to us what we need to do to get them fixed. So with that, I am going to wrap up my talk, and we'll leave it open to questions.